Hello everybody, it's Dr. Dan, and we are ready for our last day of cell culture experiments in BME 245 valve. So we're going to do our neutral red assay to see if any of the chemicals we put on the cells had any effect on their viability, cytotoxicity. So I'm going to get the stuff out of the incubator now. If you remember, I made 2 plates of cells to check. And then in the incubator, I also have our neutral red solution. Okay, so we're just following the protocol instructions on this. We're kind of at the point where sterility doesn't really matter a ton because nothing's going to really infect the cells and then live. I mean, we're really only going to do this assay for the next 2 hours, and so that's not really going to affect the cells too much. I'm still going to keep this as sterile as possible just for completeness. Okay, and the first thing we need to do with the neutral red is we need to centrifuge it. And so the protocol says that we should centrifuge neutral red at 600 RPM for 10 minutes. So I'm going to start that since it'll take some time. And so I can't centrifuge just one thing, right? I need to balance. So I'm going to go ahead and stick it in 2 different tubes to centrifuge. I should have about 24, but I'm going to go ahead and play it safe and just put 11.5-ish in each tube. We do want them to be equal so it's well balanced. And I finally found out this leaky pipette, I can kind of avoid some of this by tilting the pipette way back. I'll show you what I mean. All right, it wants to leak, but if I hold it tilted back, it doesn't get up into there, but it also leaves like a little air bubble in front. Okay, and the reason we're doing the centrifuge, if there was any precipitate, we don't want to put that on our cells. So we're going to centrifuge to make sure all the precipitate gets down to the bottom of the tubes. So I want to make sure I'm doing exactly opposite. 600 Gs for 10 minutes. Okay, so we're off and running. Okay, so now I'm going to get ready. First thing we got to do is aspirate all this media off the cells. So I'll go ahead and do that. So I'm actually going to do these bleached cells last, just so if I get a little bit of bleach on my tip, it doesn't accidentally transfer to the other wells. So I'm going from order of least concentration to higher concentration. And then finally my bleach control. Okay, now I can just get rid of this tip and get me a new tip for the other plate. So again, on all these, I'm just gonna work on a from lower concentration to higher concentration. and then again do the bleach last. That way if there's any problems, I don't have a chance to accidentally contaminate some of the other cells with things I know is going to kill it, like bleach. Okay, so now we basically have to wait. We got about 5 more minutes for the centrifuge to finish, and then we'll put the neutral red on here. While we're waiting, we can go ahead and take a look at these cells in the microscope.
So we're looking at them in the microscope at these cells. These are from the control well where we just had DMEM media, and so these cells look like they're in pretty good shape. Okay, now I moved over to look at the cells that we had bleach on, and there aren't really cells in here. This is just kind of trash left over from aspirating out whatever was left. And so that also is good news for us. That's what we expect from the bleach.
Okay, our centrifuging is about done. Just gotta slow down. Okay. I'm going to be very careful so I don't disturb any pellet that might be at the bottom. And let's look close at these. Yeah, you can definitely see some crystals down at the bottom. I don't know if you guys will be able to see on video, but there's some solid red down at the bottom that we don't want to put in into our cells. So we're going to have to be careful about that. So I'm going to get out. The way we're going to do this is just this plate again, which I love opening these things up. Whoever made these needs to put an easy open area. Okay, so I am trying to be as careful as I can. It doesn't matter as far as sterility at this point, but I just like not to contaminate things if I don't have to. Okay, and the protocol, I think, says to add 100 microliters of neutral red solution. And I just checked on that to be sure, and it is 100. Okay, so we're going to add 100 to each, and now I need to put this into our plate over here. Again, ideally, I'm not going over this. Just practicing being sterile no matter what. I'm going to be careful here not to get that little pellet at the bottom. So I'm going to start sucking from the top. I'm probably going to leave like a ½ mil in there. Something like that, just so I don't have to get it and mess with it. I'm doing this fairly slowly because I didn't want to splash it everywhere and make bubbles like that. Okay. Okay, so now I'm just going to add these 6 at a time with this thing. 1, 2, 3, 4, 5, 6. Again, my job is to make sure these are get pretty evenly on there. And here we go. All right, this is where it takes a good degree of concentration to make sure you're putting it exactly where you intend it. All the wells get filled. With the same amount of liquid. Okay. So while I was doing that, I actually wasn't going all the way down to the second stop. I was just going to the first stop so that I wouldn't make too many extra bubbles. All right, so I'm going to put these in the incubator. Now we want to do it for an hour and a half. And I will start the timer. Okay, and so next, in this hour and a half, I'm just basically going to start cleaning up some of the mess I made. I'll aspirate all that remaining neutral red out, and then bleach the aspirating flask. And I'll get ready for the second half of the experiment, which is to do the destaining step, and then measure how much dye we have in the spectrophotometer. So remember, as the cells are in the incubator, the live ones will take up that neutral red and it will be trapped inside the cells. And so then we're going to add some of the destaining solution, which is ethanol and acetic acid, which will basically burst the cells open and get all of that dye out that was trapped in the cells. And so that's a good way of estimating how many cells we had alive.